So good morning or good afternoon, everyone. It's really exciting to be here at the virtual pub for Euro-BioImaging. I really want to thank you for the invitation. This is like a really great initiative, and thanks for really building up this network that is really important for the community. So since we are in Europe, I just want to locate the lab, our lab in Europe. This is a view of Northern Europe by the International Space Station, and the Laboratory for Optics and Biosciences is located in the Paris area, not very far from the Eiffel Tower. So what do we do at LOB? So at LOB, we develop new optical tools in optical microscopy. And as you know, optical microscopy has a great compromise to image cell and tissue in terms of resolution and depth. And what we are really focusing on at LOB is mainly the development of multiphoton microscopy for tissue imaging, 3D optical sectioning, so that it is able to image 500 micrometer depths with a resolution of roughly 1 micron. And what we actually work on are also combining a different type of microscopy and nonlinear optical contrast for label-free functional imaging of tissues. So we are combining fluorescence lifetime microscopy and two-photon excitation of endogenous biomarkers. And this is the focus of today's talk. But we are also working on coherent processes like second harmonic generation microscopy to visualize collagen microtubules and tubulin for label-free, and third harmonic generation to visualize water-liver interfaces like, for example, myelin in the brain. And what we're really doing at LOB is developing these tools and applications in biomedical sciences. And we're really pushing this technology and these applications to go faster, deeper, and also to add contrast. So let's go to the outline of today's talk. So today I really want to talk about FLIM for label-free metabolic imaging of living tissues. And after a brief introduction of endogenous fluorophores, I want to give an overview of FLIM and FLIM of metabolic biomarkers and how we can also add contrast, and some of the applications specifically focusing on immunometabolism and neuroscience. So what are the intrinsic fluorophores? So most of the microscopists are usually fighting against autofluorescent, but we actually love autofluorescence and we are exploiting it as much as possible. And here you have an examples of the most important autofluorescent fluorophores that you can find in tissues. Some of them are very important, involved in physiological and pathological processes, and they have very large overlapping two-photon absorption and emission spectra. And today we're going to focus on NADH and FAD because those are the ones who are involved in metabolism. As you know, NADH and FAD are the primary electron acceptor and electron donor in oxidative phosphorylation. And the reduced form of NADH is fluorescent and is emitting in the blue, and FAD is also fluorescent, is emitting in the green, while NAD plus and NADH are not fluorescent. So since the pioneering work of Britton Chance, people started measuring optically the redox ratio by measuring the ratio of FAD divided the sum of FAD plus NADH, and then evaluating how this is contributing, how oxidative phosphorylation is contributing to the metabolic state of the cell. So in two-photon, what you have to do is you can use a tunable laser. You can excite NADH around 750 and collect with a blue filter, and 900 for FAD excitation and collect in the green. So by looking at these two biomarkers and by intensity redox ratio, again, as I said, we can have a first evaluation of how much important is oxidative phosphorylation and respiration in the mitochondria with respect to glycolysis in cells and tissues. And here I just want to point out some very nice reviews that are covering the intrinsic biomarkers in tissues if you want to know more. So, but why we are doing FLIM? So we are doing FLIM because if you only take in account intensity, you might have a lot of problems and biases, especially in tissues and scattering tissues when you go deeper in tissues. While lifetime is much more robust because it's independent on the concentration of the fluorophores and is much more robust in terms of heterogeneity and scattering. So that's why we do fluorescence lifetime microscopy, and we measure the lifetime of fluorophores, because the lifetime of fluorophores, it depends on the molecular structure of the protein or the fluorophore, but also and especially is very sensitive to the microenvironment through the non-radiative decay. So FLIM and the lifetime decay can really report on binding, quenching, pH, viscosity or energy transfer. So before talking about the metabolic imaging with FLIM, I want to tell you what is FLIM for people who don't know. You can do the FLIM in the time domain or in the frequency domain, but in the time domain, you can do time correlated single photon counting, that is also called TSPC cards, and you need special electronics to do this. And what you do is like you measure the arrival time of every photon with respect to the laser pulse, and then you measure this time, and then you build your statistics and your multi-exponential decay. So as I said, you need special electronics. There are some companies that are providing electronics. Those ones that I'm showing here are the ones that I actually personally use to perform FLIM measurement throughout my career. But if you want to know more about instrumentation and how to do this, you can refer also to these very nice reviews that are recently published. So here at LOB, we develop our own electronics. We have commercial electronics, but we also develop our own electronics developed by Xavier Solinas. And this is like a fully programmable electronics based on FPGA. And we can perform a FLIM with these electronics, but we can also, for example, multiplex, unmix different signals with different polarization. So, so I want to also talk about data analysis in FLIM. So the classical way of doing data analysis is curve fitting in a multi-exponential decay. This can be like pretty complicated and also very slow. So that's why we mainly focus, since my postdoc at the LFD with Enrico Graton, I really mainly focused on fit-free analysis based on phasor analysis, because it provides an unbiased representation, and of course is much faster because it doesn't require any fit. So as I said, the phasor approach to fluorescence lifetime microscopy has really been established by Enrico Graton from the Laboratory for Fluorescence Dynamics, where I did my postdoc. And in this seminal paper by Michel Digman and Enrico, they were like really explaining how to do phasor from analysis from the time domain or the frequency domain. So if you are in the time domain, you need to transform your data with a FFT. And then you can plot your decay in 2D plots, which is called a phasor plot. For example, phi, if you have a very short lifetime, you're here with a phi phase shift very short, while longer lifetime are along this universal circle. So what are the rules of phasors? So the phasors, so if you have single exponential, your lines are going to be here along this universal circle, while if you have multi-exponential decay, everything is going to be in the middle. I'm actually going to activate my pointer finally. As I said, the multi-exponential decay are inside the universal plot. And what is interesting about phasor is the linearity of the phasor space in terms of non-interacting fluorophores. If you have fluorophore blue and green, regardless the complexity of the decay, you can always quantify very easily their contribution just with the graphical interface, the graphical measurement. So this is actually very simple with respect to multi-exponential decay. And this is, for example, like the fingerprint of the most important intrinsic autofluorescence biomarkers that have been measured. We measured the lifetime, and then they have a specific location in the phasor part. For example, here you have free NADH at 0.4 nanoseconds and bound NADH, which is multi-exponential. And then we use this as a map and a guideline to interpret our data, just keeping in mind that the references in solution can be only a reference. And when we go in cells, in tissue, we need to have more robust calibration because the pH and the viscosity is changing. So if you want to do analysis, we develop with Phaser, we develop an open source software, which is called FLUT, Fluorescence Lifetime Ultimate Explorer. And this is a Python graphical interface. And we developed this open source software with the goal to have something very user-friendly and to be able to analyze larger data sets. So the code and the executable are available on a GitHub page and are free and open source. And then you can analyze all and reproduce all the data that are deposited here that are represented in the paper. And Bahar Asadipur also gave a very nice workshop and tutorial on the I2K conference that you can watch online if you want to learn more. And we'll keep developing FLUT with the FLIM team now, and particularly with Xiaotong, who joined the lab recently. So the goal of FLUT and open source data is really to analyze FLIM in the time domain. And we are able to analyze FLIM from the most important FLIM cards that are commercially available. We need just to transform the data in a TIFF format. And I just want to go on the main goals, focus on the main goals of why we're doing open source software. And actually, we want to do open source software, especially for FLIM, to have reproducibility and have open access for everyone and to make easy and quantitative analysis. So once we developed FLUT, we were actually the first to do open source code in Python, but since then there are many other open source initiatives like PhaserPy, Napari FLIM, PhaserPotter, and Phaser Identifier and FLIMPA in Python. And this is actually great for the FLIM community, and there are also synergies and collaborations that are opening up. And I want to stress again the importance of open source software for microscopy, in this case for FLIM. And we used FLUT and FLIMFit to analyze data for metabolic imaging in these consensus guidelines for label-free optical metabolic imaging, which is like initiative led by Irene Gorgakpuri to ensure accuracy and reproducibility in metabolic profiling. So again, open source software is very important. And then I want to move on the focus of today's talk. So how can we do FLIM with metabolic biomarkers? And how can we check the connections with metabolic pathways? So as I said before, we're really focusing on NADH and FAD because they are like really the acceptor and donor in the oxidative phosphorylation. But if we do fluorescence lifetime microscopy, we're going to be much more precise. And this is because the lifetime of NADH and FAD are very sensitive to the binding of those fluorophores. So if NADH is in a free form, it has usually a 0.4 nanoseconds, while NADH in a protein-bound conformation has a much longer lifetime, between 2 and 3 nanoseconds. And what is actually very interesting is that the protein-bound NADH lifetime depends on the enzymatic binding in the TCA cycle. So on the contrary, FAD has a much shorter lifetime when it's protein-bound and long lifetime when it's free. And so by measuring the lifetime of these fluorophores, we can really measure some, obtain information about different metabolic pathways like glycolysis, oxidative phosphorylation, but also a fatty acid synthesis, oxidation, and oxidative stress. In the case of oxidative stress, we are mainly talking about NADPH, which is of course different from NADH, but I just want to point out that these two fluorophores are spectrally identical or very similar. And so we mainly talk about NADPH because you are collecting signals from both of them. But if you just go back and focus on NADH itself, or NADPH, again, so when we measure the lifetime of free NADH, we can measure, basically we can quantify through multi-exponential fitting or phasor analysis, the fraction of bound NADH with respect to free NADH, which is actually proportional to the redox ratio NAD plus divided by NADH. And in this way, we can quantify the contribution of oxidative phosphorylation with respect to glycolysis. So typically, if a cell undergoes mainly with oxidative phosphorylation, we have a long lifetime and more bound NADH, while if a cell is glycolytic, we have short NADH lifetime and more free NADH. So again, so just to remind you, this is label-free. We don't need to add anything. We're exploiting the intrinsic autofluorescence of NADH. We have subcellular resolution. So here you can see, for example, very bright mitochondria with very long lifetime, NADH lifetime, and bound, while in the nucleus we have dimmer signals and shorter lifetime. So here is just a selection of some of the papers that we recently published since my postdoc and also here at LOB. And I just want to point out that, of course, we want to go on this tissue scale and map metabolic gradients in tissues. And here is one of the first examples where we could measure metabolic gradient associated to stem cell differentiation inside a small intestine. Yes. So again, so what are the advantages of FLIM with endogenous biomarker? As I said before, we are on a subcellular level, so we can see what is happening inside the cells, so the mitochondria, cytoplasm, and nucleus. And this is label-free, so non-invasive. We don't need to add any marker. And of course, this is, has a big difference with respect to mass spec or like, for example, flux analysis, where we have to lyse the cells or like see, we are limited in terms of spatial resolution. So actually, we think that FLIM of endogenous biomarkers could also help to understand the other types of metabolic imaging, like fMRI in the brain or in other organs, to understand what are the contrasts and what is the source of metabolic changes at the level of the cells and subcellular level. So before going to the application, I just want to give you some relatively technical details, but these are very important because these are considerations that you have to do when you start to do a measurement, and then you have to really think carefully about your applications. So when you do FLIM and especially metabolic imaging, you always have a trade-off between the number of photons, so basically the desired resolution you want, the photon flux, so how much power you and how much light you shine on your sample. So that has been effect on the pileup and phototoxicity, and also the acquisition speed. So what is the dynamic of your system and how fast you want to do the imaging? So what are the rules that I suggest? The first one is to, first of all, determine what is the desired number of photons. For metabolic imaging with NADH, we usually use in the order of 500 photons for phasor analysis. Then in the second part, you need to know what is the maximum photon flux that you can use with your microscope, with your electronics. And then you need to take care of the power. So you need to use a power that is low enough in order not to induce damage, which is very important, especially if you want to measure metabolism. And then at the end, you need to acquire enough time to have enough photons. So I just really recommend to put all this in your publication so people can reproduce your experiments. Everything is clear. So if we go on the number of photons, the single photon counting process is usually Poisson distributed. So the error squares, so it depends on 1 over the square root of number of photons. And the rule of thumb is that for, let's say, a multi-exponential fit, we use like 1,000 photons, but with phasor analysis, we use less photons, like around a few hundreds. And this is just to give you an idea what is happening in the phasor space. If you have increasing number of photons, our cloud is kind of shrinking, and then your measurement is going to be precise. Of course, both in the single exponential case or in double exponential decay, where you have free and bound NADH. So the second point is you need to know when you do an experiment in your, for example, microscopy platform or your microscope, you need to know what is the dead time of your electronics and your PMT, because this is going to determine how fast you can go without having artifacts or what are so-called pile-up effects. Because with FLIM cards that are relatively slow, the dead time is long, and so you lose photons, and then you can just create a bias in your measurement. So recently you have FLIM cards that are being developed with shorter dead time. And so with these shorter dead time cards and hybrid detectors, we can just go faster. But, so the bottom line is that you need to know what you have. So the third point is adjust the power in order not to induce photo damage of the cells. So the rule of thumb is that for longitudinal imaging, we use less than 5 milliwatts to follow like the same cells or the same tissues. But you really need to consider like the bleaching, but especially the damage inside cells. So what do you have? So you can evaluate your photo damage, for example, with red viability dye, like we have done in this publication. But I just want to stress that the main limitation in metabolic imaging and FLIM is actually the power that we can use without inducing any damage and photoperturbation inside the sample. This is because NADH is pretty dim and it has a low quantum yield. And then finally, so the fourth point is just that you need to stay there and acquire enough time in order to have enough photons, the photons that you want. But just to give you an idea, we usually acquire roughly between 100 and 200 microseconds in order to have a good number of photons, 500 photons, with these conditions that I was explaining. So finally, I just want to point out that here we're doing label-free FLIM of NADH, but this is actually compatible with other markers and especially with red fluorophores or red fluorescent protein, because you can excite them simultaneously and collect them in two different spectral range. And then we use this a lot to do, for example, identify cell types or subcellular compartments. So I want to talk now about the metabolic pathways. How do we know what we're measuring? Because the metabolic pathways are many and also very complex. So in cells, we need to do calibration with metabolic effectors and metabolic drugs. What we usually do is, for example, we inhibit metabolic pathways. For example, here we inhibit electron transport chain with potassium cyanide, and we see the movement of NADH toward the free NADH position. Here we induce oxidative stress, and then we have an opposite direction. And then when we just really focus on the glucose levels or inhibition of glycolysis, we also can characterize really the trajectory of glycolysis and oxidative phosphorylation in the phasor location. So these calibrations in cells are very important, and I really suggest to do them every time. And then here you have some open access data, published data that have been analyzed with FLUT, with metabolic drugs that is inhibiting oxidative phosphorylation. If you want to play with the data, you are welcome to do. And I just want to point out that there are also new biomarkers for oxidative stress and lipid-associated fluorophores. And we also have done some automated segmentation of lipid droplets and by using this long lifetime. And I just want to point out that NADH lifetime is also changing with fatty acid oxidation and synthesis. So the changes that we observe are not unique, so to one metabolic pathway. So in order to identify in a unique way the metabolic pathway, we cannot only look at one biomarker, but we need more biomarkers. And so that's why we want to add contrast and have a look at multi-parameters. So in the recent years and recent publication, especially by Melissa Scala and Irene Gorgakudi lab, people and us also, we've been showing that these kind of biomarkers are very complementary. And if you only look at NADH, it's not sufficient to discriminate all the metabolic, for example, fingerprints or like, let's say, phenotypes of cells. But by adding also the FED lifetime and the optical redox ratio, you can identify metabolic pathway more precisely, and you have like much better information. And in order to do this, and in two-photon microscopy is actually relatively complicated. And this is because to excite NADH and FED simultaneously is not very easy. So in collaboration with Emmanuel Boreper, Pierre Mahou, and Lin Ung, we developed a new wave to do simultaneous excitation of NADH and FED with wavelength mixing. So wavelength mixing has been first used for brain-combo imaging, and we adapted this with a commercial laser source with a fixed output at 1040 and a tunable output at 740, and we mix the two beams in time and space. So if we only use 750 excitation, we mainly excite NADH, but when we mix the two beams, we create a virtual wavelength around 900 that is able to excite FED independently on NADH, and then we can basically excite NADH and FED simultaneously, but in a very efficient way, so with a lot of photons. And this is, of course, very important in very dynamic systems, like, for example, this is an example of developing zebrafish embryo. So what we can do with wavelength mixing, so we can excite NADH and FED, and we demonstrated this in a collaboration with Ana Maria Pena's lab at L'Oréal to look at reconstructed human skin tissues. And here we demonstrated the first one-shot NADH and FED intensity and FLIM imaging in tissues. And we were able really to map metabolic gradients that are associated to stem cell differentiations in the skin. And we actually applied this methodology to investigate how these two biomarkers are behaving in the reconstructed skin when we shine the screen with long UVA and induce metabolic stress. So in collaboration with L'Oréal, so we use a UV lamp and specific physiological doses like 25 and 40 joules per centimeter square. And these doses are not changing, leading to any morphological changes at 2 hours. But we perform the imaging over time at 30 minutes and 2 hours after the UV light exposure. And we can image both the basal layer and the dermis. So when we expose this UV, so the reconstructed skin with UV light, we see an increase of the lifetime of NADPH and also of FED. And we think that this is actually due to the oxidative stress that is induced by the light, the UV light inside the cells. So we fully characterize this in the epidermis, so and also the dermis, so in different types of keratinocytes and fibroblast cell types, and also with different doses and of different times. And basically what we found is that the take-home message of this work is that NADH and FED are like giving a different response. So the metabolic response is biomarker dependent, but also cell-dependent and dose-dependent. So NADH biomarker shows an early modulation in fibroblasts, but not keratinocytes. And FAD biomarkers showed an early modulation in both cell types. And FAD is also sensitive to the UVA dose. So yes, so in the remaining time, I would like to show you some other applications that we have been working on, and the field of FLIM and metabolic imaging can be used for, especially for immunometabolism and neuroscience. So as you know, there is a tight connections between metabolism and function in different physiological and pathological processes. And here, first, we're focusing on immunometabolism. I'm going to give you some examples across scales and some examples in the fast metabolic switch in T cells and metabolic switch shifts in tissues. So in a very, let's say, binary vision of immunometabolism, you have M1, M2 macrophages, so the pro-inflammatory macrophages that is leading to like a Warburg effect and more glycolytic, let's say, metabolism, while pro-generative macrophages are, let's say, less glycolytic and more oxidative phosphorylation. But we all know that the reality is much more complex, and then we have really a big heterogeneity and a big variety of metabolism. So metabolic imaging and FLIM of NADH has been used extensively in this field of immunometabolism by many labs, in particular by the lab of Melissa Scala, to study the immune cell activation, to perform cell classification, but also characterize single cell heterogeneity analysis in time and space, and also to design and improve the CAR T cell generation. So there are many also applications in living tissues and engineered tissues that are coming up in the field, and some applications are also popping up in brain, in vivo brain, in the brain of the mouse and in the zebrafish. So I want to tell you what we've been doing in this field of immunometabolism. And this is a collaboration with Claire Rose and Noémie Paillon. And this is a work done by Lin Ung. And in this paper, we really measure the fast metabolic switch in T lymphocytes, in T cells, by label-free and by performing longitudinal imaging of the T cells. And then we were simulating our immunological synapse by coding cover slip with activating antibodies. So we were doing longitudinal imaging in 3D. We performed a single cell segmentation in 3D. And what we observed is that we have metabolic shift. So we can track the single cell. For example, here is like a cell over time. And in activated cells, we have a clearly metabolic shift toward glycolysis that is in agreement with other publication. But for the first time, we were able to measure like that fast metabolic shift right after 10 minutes of activation. So I just want to point out that this is in a Jurkat cell line, which is like a stable line. And this is also increased with area, is correlating with the area increase of the cell. And then if you look at like also the three-dimensional distribution in the cells, we see that we have an asymmetric distribution of the metabolic profile in these cells. And in particular, close to the immunological synapse, we have a longer lifetime and more fraction of bound NADH. With respect to the other side. And I think we think that this is really due to the cell polarization, so to the fact that mitochondria are migrating toward the immunological synapse in activated cells. So we proved this also in primary cells. I just want to point out that in primary cells, we also have like a glycolytic shift in the T cells. And although we have more variability, this shift is present very clearly. So we also are we're imaging a metabolic shift in tissues, and this is the work in collaboration with the Paris Brain Institute and Bruno Stankov team. And this is actually a work done by Bahar Asadipur, and she measured actually a metabolic shift in microglia. So in microglia, when we have pro-inflammatory microglia in demyelination context, we see metabolic shift toward a glycolytic phenotype, so shorter NADH and like a rounder shape. So now I want to give you some overview about some other applications of metabolic imaging and NADH FLIM in neuroscience. And in particular, I want to give you an idea of what can be done across scales, and 3 applications that we have been working here at the LOB, in particular on neuronal differentiation, neuroglia coupling, and also memory formation. So also neuroscience, metabolic imaging and FLIM NADH has been used to measure metabolism at the single cell level and subcellular level, but also the tissue level and engineered tissues, and a lot also in vivo. So for example, in the brain of mouse, zebrafish, and also Drosophila brain. So, and the applications are really to study neuroglial coupling, neurodegeneration, brain activity, and also memory. So I'm going to give you an update about what we've been doing at the LOB, and in particular during neuronal differentiation. And this is a long-lasting collaboration with Lorena Aguilar Arnal from the University of Mexico. And with her, we've been measuring the metabolic fingerprint and mapping metabolic maps of cells during differentiation into neurons. And coupling this also with RNA sequencing to really understand the connections between epigenetics and metabolism. So Xiaotong, who is working now in this project, is developing new fast FLIM methods for metabolic imaging, and with the goal of mapping tissues, entire tissues like zebrafish, and during differentiations, and also brain organoids during the neuronal differentiation and the neuronal lineage. So we're going to use also genetic engineer approaches to really understand what is the link between the metabolic transition to transcriptional reprogramming. So what we have been doing so far is that we are able to measure metabolic shifts during neuronal differentiation, and we can characterize cellular and subcellular heterogeneity. So in embryonic stem cells, human embryonic stem cells, neuronal progenitor, and neurons. So these are dopaminergic neurons. And I just want to point out that this technique we demonstrated that can also distinguish neuronal progenitor and glial progenitor in mouse cells. And we're really trying to understand and characterize the lifetime with different biomarkers in the phasor space to find a unique really fingerprint for like a differentiation stages of cells. So we've been pushing this technology to map in vivo in the zebrafish all the metabolism in the entire zebrafish, but with a specific focus in the brain. And then here is a zebrafish at 3 days of post-fertilizations, and you can clearly see the metabolic heterogeneity inside the brain. Some regions are very different, and you can see heterogeneity also at a single cell level and at the level of axons. So we can map without any markers, so the spatial and temporal patterns in zebrafish during development And then we see that we really have a very, very big increase of lifetime of NADH that is showing an increase of OXPHOS and a decrease of glycolysis during development. So in the current work, we are focusing on dopaminergic neurons in the zebrafish in vivo. And we are also imaging brain organoids with FLIM and without any biomarkers, because these brain organoids are done with from the human embryonic stem cells. And this is a like ongoing project now here at LB. So another application I want to talk about metabolic imaging in neuroscience is in the context of neuroglia coupling. As you know, the metabolism is very important in the neuroglia coupling in the context of brain activity and myelin plasticity. And this is actually one of the bases of functional MRI and is not completely understood. So there is like a shuttle of, for example, of metabolites between astrocytes and neurons during activity. And this is the pioneering work of Wet Lab that measured for the first time in vivo the astrocyte neuron lactate shuttle by looking at the NADH intensity. And after neuronal simulation, you see first an oxidative metabolism in neurons, and then later glycolytic metabolism in astrocytes. So what we're doing here at LOB in the brain, so Bahar Asadipur during her PhD has been measuring NADH lifetime intensity and lifetime in the organotypic slices of cerebellum. And in the context of brain, so myelin formation, so we do THG imaging to probe the myelin distribution and NADH two-photon excitation to map the heterogeneity of NADH intensity. So with lifetime, we can measure the lifetime of NADH in every pixel of the image. And then by using red reporters, we can do single cell analysis and see what is happening inside different types of cells. And what we observe is that we have very different metabolism and very different lifetime in neuronal Purkinje neurons. Purkinje neurons have the most oxidative phosphorylation life and long NADH lifetime. That is actually goes along with the need of a lot of energy and activity of the neurons. And the microglia are actually more glycolytic, and also oligodendrocytes are pretty glycolytic because they are like need to produce biomass very fast. So an interesting perspective in this field would be if there is a connection, and can we measure like connections between metabolism and neuronal activity, depending on what type of neurons. And actually, this is like a recent collaborations with Pierre-Yves Placé at the SPCI. And our collaborators, so we're able to image the Drosophila brain in vivo and map the metabolic states of the entire brain or most part of the brain. And what they were able to do is to see that different subtypes of neurons have a different metabolic basal profile. And some neurons, so the neurons that are involved in middle term memories, we observed like a metabolic shift during memory formation in these mid-term memory neurons. So to finish, I want to highlight some other applications in the field. I cannot cover everything because it's too much, but I just want to highlight what can be done what the community has done with metabolic imaging in cancer. And of course, metabolism is very important in cancer. There is a metabolic shift in different types of cells, cancer cells and immune cells. And so FLIM of NADH has been of a great help to characterize metabolic phenotype and heterogeneity in single cells and also at the subcellular level. And FLIM can be used really to sort cells and identify cell types and metabolic phenotype. But in patient-derived spheroid and organoid is also very used to also characterize metabolic association to motility and migration and metastasis. And of course, if you go to a larger scale, intravitreal imaging or in vivo diagnosis, you can image the entire or like larger area, or with a mesoscopic approach developed by Laura Marco's lab mainly. You can also delineate, for example, tumor borders. I just want to highlight, this is label-free, but we can also add probes, as I showed before, red probes. And in the context of cancer, what is actually very interesting, you can add, for example, probes to measure oxygens, and you can perform metabolic imaging at the same time of oxygen concentration measurements with Fluorescence Lifetime Microscopy. And this also is a very exciting field in cancer metabolism. So to conclude, I hoped that I showed you how can we do a fluorescence lifetime microscopy with intrinsic biomarkers, especially with NADH, and how this is very powerful for metabolic imaging because this is labor-free, non-invasive, and can report on oxidative phosphorylation glycolysis balance in tissue and cells. Of course, you really need to think carefully about your applications, about your microscopes, and really balance between light dose resolution and acquisition speed. And we showed like some examples of multiparametric imaging of NADH and FAD, how these two biomarkers are sensitive to different pathways, and then how complementary can be. We demonstrated how metabolic shift, measured a metabolic shift in immune cell activation, stem cell differentiation, and also in larger tissues of different types. As perspectives, I want to highlight, to me, one important thing in the field is that we need to work still a lot to define what is the sensitivity of these methods to metabolic pathways, and by also, for example, using mass spec measurements or different types of measurement, and implement this for longitudinal metabolic measurement in vivo. And of course, this will be very important for fast FLIM of metabolic imaging. And then last but not least, I want to highlight that this NADH FLIM is very compatible with other nonlinear type microscopy like second harmonic generation and THG. And this is what we actually working on a lot on the LOB. And to conclude, I just want to give a perspective on fast FLIM. So here there are like three recent reviews on fast FLIM. Now in the field, there are many different technological advancements to push FLIM to go faster in terms of hardware and microscopy and applications. But as I said before, the main limitation in classical time-correlated single photon counting is that the electronics is slow. So there are new electronics, but there are also, to go even faster, you can just think about parallelizing the collections, so parallel units of parallel detectors. And really, you can always see an improvement from classical time-correlated single photon counting, hybrid detector with fast electronics, and then you can start parallelizing. You can really gain in terms of time much more. And for parallelization, you can use, for example, ICCD intensified camera for time gated FLIM, or SPAD arrays that now are developing also in larger arrays. And with all this technology, we can also do, for example, light sheet FLIM. And the question is, are these fast FLIM advancements applicable to metabolic imaging? And the answer is yes, but this field is very challenging. There have been some papers really using this technology in metabolic imaging, but still is very open as a field, and also challenging because NADH is very dim, and you always have to balance all these three: resolution, light dose, and acquisition speed, especially for metabolic imaging. So to conclude, I want to thank all the people who worked at the Laboratory for Optics and Biosciences on this project, and in particular Lin Ung and Bahar Asadipur, who basically produced all the work that have been I showed today. I want to thank also Emmanuel Boreper, Pierre Mahou, Xavier Solinas, and Xiao Tongguan. And I want to thank all my collaborators for these exciting collaborations and exciting science we've been doing in the last few years. And I want to thank the funding, and thank you for your attention, and see you around in Europe.